Yeah.
Yeah, I started recording.
Okay.
All right, guys. So
So we're gonna start with the last author, right? So W. A. Leo Smith.
yeah, if anyone want to jump on for the last author, that'd be great.
So I found that Leo actually goes to the University of Kansas. It seems he specializes in evolutionary biology as well.
Yeah.
And according to Google Scholar, he does have a lot of publications. The same thing goes with Matthew. They both have a lot of publications, and they both, especially Matthew, he seems to really specialize in the deep sea. A lot of his publications are in the deep sea and stuff.
Yeah, that's definitely what I saw too. On based on his website, it looks like he specialized in the biodiversity of the deep sea,
Hmm.
and he also focuses on the um I don't know if you guys remember, but the ideas of comparing DNA and how you do like phylogenetic hypothesis
Hmm.
and all that kind of stuff.
Yeah.
So I think that's what Matthew is really into.
Yeah, you know, he specialized in deep sea. I actually see some publications on on freshwater fishes as well. Like he focused on
Yeah,
cichlids.
many things. Yeah,
Yeah, a lot of
like
things.
anything from like estimating like the divergent time, diversification rate, whatever. Just Matthew does it all.
Yeah.
Yeah, I mean, he even had an exhibition at a museum about bioluminescence specifically.
Mhm.
I
Yeah,
mean, you could see one of the pictures. Yeah, he seems to be pretty knowledgeable
yeah,
Yeah.
in the field.
On
mhm.
his website, you can see like some of the fishes I guess he has studied,
Hmm.
Yeah.
and
They
they
all
look pretty
very
cool.
they all very similar to this paper.
Yeah,
Yeah.
exactly.
Yeah.
All right, and then with this paper, from what I think or from what I get in this paper is they just want to see the functions of the I don't know bio how do you say that word?
Luminescence?
Biodiversity.
Biolum
Bio,
Bioluminescence.
yeah,
biolum
bioluminescence, whatever that is. So they from what I get, I think there's two functions to that one. One of it is to predation, and the other one is for like I don't
communic
remember exactly, but there's another functions that helps them to diversify at a much faster
it's
rate.
mostly communication, right? Like
There you go.
they
Yeah.
can diversify within each
Uh-huh.
other, like by communicating with
Yeah.
their
And that's how they come to
bioluminescence.
like at a faster rate diversification.
Yeah.
Yeah.
All right.
Yeah, because it uses bioluminescence to communicate, and it also helps them defend against predation too, or find or attract prey.
That's like the initial I think I have it on here. Let me
I mean,
go.
and that's also the research question, right?
Yeah.
Yeah.
They want to find out how they diversify,
Definitely.
if
Yes.
it aids in diversifying more,
Yeah.
or
Oh, wait.
if it's a factor in diversifying, I
Right
should say.
here it says, well, I wrote that there's like four main uses of bioluminescence, and it was
Okay.
camouflage, defense, and predation and communication.
Yeah, but
Hmm.
those camouflage predation, I think those are one category, and then
Mm.
Oh,
communication is
It's
two
just like a
is
whole
is
other one.
another one.
Yeah,
Another category.
But
Yeah.
then
yeah.
I don't know if you guys remember, there's also the lateral and then the ventral.
Is it the
Um,
lateral phonophores or something?
yeah, because
Yeah. Uh-huh.
some fishes have it on the side, and then some fishes have it on the bottom of their body,
Oh yes.
and
mm-hmm.
the locations of those bioilluminants determine
It's where, yeah.
functions
Oh,
of the yeah.
okay.
I saw it. Yeah, I remember seeing that in the paper.
Yeah.
Yeah.
Yeah. Okay. Should we start going over the
The
figures then?
figure.
Let's
Yes.
go ahead
How
and
about
take
the
a look.
material method?
Oh, okay.
So, I mean, first of all, I mean they mentioned that the main one was tracing back the phylogeny, essentially, right? That was one of
Yeah.
the main ones that they wanted to do.
That evolutionary history, of course,
Mhm.
that one, yeah. Um, what else?
Well, first it was like the temporal phytology, and then it was like the six fossil calibration,
Yeah,
and then
yeah.
they did like sequences aligned with the MAFFT, and then they did like the beast versus like 1.75 to determine the topology and reconstruction relative to divergent time.
Definitely, yeah. If we want to take a look at the diagram that I did
Mhm.
for the material method, but I don't know if this is 100% correct, but definitely I try my best to put everything together.
See.
So I understand the very last part for sure. The last method that they did is basically to just use a regular Canon and photograph the exact locations of the bioluminescent.
Mhm.
What is it called? It's called the
Bioluminescence.
photophore. Is
Yeah,
it called
the
Photophores,
photophore. Yeah,
the
photophores. Yeah.
photophores?
photophores.
Yeah, they just exactly take pictures of it and then they run it through the test, whatever this test called, the software TPSD something, and
Yeah.
then those tests is how they get these graphs right here, and that's just how they compare.
Just to compare between each other.
Yeah, like um the lateral, the ventral, and so on and so on.
Yeah.
Yeah, we'll go in details on those
Mm,
graphs later on.
yeah, we can
Yeah.
go one by
Mhm.
one.
Mhm.
Should we? Okay. Should we just start with the looking at the tree in depth?
Yeah, let's go ahead and do that.
Yeah.
Right here. There we go.
So this artist says we specifically look at clade G, right? The one that's marked in green.
Uh-huh. Hmm.
Yeah.
Yeah. I mean, and then all the letters on the left side, the fishes, the ones that are labeled with the fishes on the left side of
Yeah.
that.
Yeah.
Um,
That's the
that was just to look at the. I'm not sure as to what that was looking at. It was just basically what was that looking at?
it's
The
like
figure one. You know how when
taxo. Wait, which one?
well the fishes
Fishes?
that are right above, yeah, figure one. Basically all those fishes that are labeled right there.
Oh, that's the taxa on the phylon.
Is it on the clade? The ones that are on the clade, basically.
Yeah.
Yeah. Because you can see A, B, D, E like
Mm.
marked on the clade.
Yeah.
Oh, I see.
Yeah. So those fishes were like located there.
Mm,
Because like, or else it would have been too like crammed inside.
Yeah.
I see.
At my note right here, which one are we looking at right now? Is it the
The one
tree?
on the
We
left.
were just looking at the fishes on the yeah, like
Okay.
right there where the arrow
All
is.
right.
Oh.
And then for the graph, what I got, it's just x-axis is just the times or generations, and then the y-axis is the species richness.
Mhm.
And I also define species richness, which is the numbers of different species in a given area or
Or the
in,
community.
or ecological community. And then from what I got in this graph, it looks like the ventral photophore fishes, they have evolved for such a long times compared to the newly ones, like the basically like the we comparing the ventral and the lateral, right?
Yeah.
And it looks like the lateral have evolved at such a like more recent compared to the ventral.
Yeah, like the venture's more. I'm sorry, more like older, essentially, right? Because
Yeah,
looking at the
like as
millions
you
of
can see
years,
right here,
yeah,
like right here, this is would be like the ventral would
that's
be
venture.
around on their belly, and then this is would be like
On their sides.
the lateral,
Yeah.
which is on the side. Yeah,
Yeah.
and we can see it'll be like much more recent.
Yeah.
Yeah. Okay. So anything else that you guys want to add?
I mean, and that thing is also saying the graph is also saying that there's more species that just ventral alone is more rich in species because that 95% confidence interval is that saying that basically more fishes fall under just ventral. rather
Definitely.
than okay.
Yeah.
Okay. We can go ahead and move into the second one then.
Right here.
So these are already looking at the results.
Yep.
Yeah, this is already part of the results.
So even just so the first figure talks about the patterns, right?
Mm-hmm.
Yeah.
Isn't it the lateral photo for locations for specific to its species and identifies its species?
I mean, it's species specific, right? All the basically they're part of the family of the fishes that they
Uh-huh.
have their specific points of bioluminescence.
Yeah.
Yeah. From here, I wrote a note down. I just put it describes the patterns of the specific patterns of the photophore, like the lateral one.
Okay.
So we can see they have this specific pattern right here, and that is for the subfamily Lamphani tiny.
Lampanticinae. Close
That
enough.
one.
Mm-hmm.
All right. And then this one right here, they do it for the ventral position, and we can see they have like a specific or pattern for that ventral photophore.
And it shows on the fish too, right?
Yeah.
Yeah, I
Yeah.
mean, you
It's the
can see the
highlighted
points.
ones, right?
Yeah, you can see the points where it's actually.
Yeah. So to summary, these two graphs right here, it's both of them describe the positions of photophores of the same fish, but then for two different positions, ventral
Yeah.
and lateral.
Yes.
Yeah, and then they do the same for the next fish for this.
Yeah.
They showed also the patterns and the locations of
So,
the
yeah,
photophores. Yeah.
Yeah.
One second, guys. So I noticed that there's a key right here.
Mm-hmm.
Does that mean they also compare the position to different other species too?
Yes.
Like
I
the color
think
code right
so.
here?
Yeah, they
Yeah.
were
It
they were
looks like
already
it.
knowns, right? Basically, they
Okay.
gathered those from
Other
the
research.
already existing
Yeah.
already existing
Okay.
research papers. I mean, and it's
Uh,
it's a good comparison kind of to see.
Yeah.
Yeah. Yeah.
It could give you a
Definitely.
better idea on who relates to who a little bit better.
Yeah.
But yeah, I mean, I feel like it's a very
And then
understandable graph.
definitely.
It's very
And
clear.
I think I'm not sure, but let me know if I'm wrong. But I think the how they did this or how they generate this data, it's basically take pictures and then they run it through that software and then it generate them like an algorithm or something. And
I mean, they use a lot of terms that are kind of
Unknown to us.
yeah. I mean, you really
yeah,
gotta look up every single one exactly to
but
really
definitely we know
understand.
it's generated by a computer, right?
Yeah.
Yeah,
Yeah.
this is definitely
It'd
computer
be too hard
generated,
to
but
do
Yeah, it's
in
not hand
person.
plot.
but it's a lot of terms that for our knowledge as of now, it's difficult to understand still.
All right. Okay. All right. So these species right here.
Yeah. It's just showing the same thing, right? The lateral and ventral patterns, where the locations are.
But for different species now.
Yeah.
Yeah.
Okay. Yeah.
Does it relate either one, like just based on the graphs alone, are they related? I mean,
What do you
they
mean?
seem kind of like because they're different fish, right?
Yeah.
They seem almost like not exactly the same, but very near the same positioning
Oh,
as like the
I.
ventral and the lateral,
Oh, where
You
the
mean like?
where
Go
the
ahead. Go ahead.
bioluminescence is. Yeah, like it seems like they're not the same, but like they could.
well, on the fish, I feel like they look like they're at the same around the same position, but then
But
looking
not
at
the
the actual graph, like they don't look that similar.
yeah.
Yeah. Because if you look at the graph, there's like different patterns all over the place,
Mhm.
or
Yeah.
photophores all over the place. Like it's not actually really close. Like you think it is.
I see.
But one thing that we can get from these graphs is for sure some of them overlap.
Yeah.
Yeah.
For
Yeah.
sure.
That's for sure. Okay, and then let's move on to the next figure. And again, the one more thing, the how they got this data is again just taking pictures and then run it through a software or something like that.
They'd run it through Medusa, or
I think so. We know it's a software, but
yeah.
all right. So okay, so this one right here.
Oh, right.
Oh,
All right.
This one is just kind of this one showed the relatedness. This is where the relatedness came from. Basically, that it was showing, I believe, from what I got, is that the
top.
the micro the which clay doh? Let me remember the name because they're really
The
freaking
top
long.
one or the bottom one?
The bond one, the graph one,
You're
the
talking about the graph?
the one with the
Okay.
blue, yeah.
Yeah.
The make the Friday clade. Oh shoot! Give me a sec.
Yeah. Mm. I
It
got
was
that most
basically
of that.
it's basically the bioluminescence trait is derived from that clade because if you scroll up, let me find it.
Which one right here? Where?
Because if you take a look at the A,
Uh-huh.
Uh-huh.
Yeah.
that basically bioluminescence was derived from
Oh,
that clade.
Yes.
Is what I understood. And then because in this clade is where the lanternfish, the Myctophidae and the black chin, which are what are being, those are the two fish that are being looked at. That's where they derive from, which is what all this graph is basically showing. Or not the graph, sorry, the tree.
yeah.
Yeah, I think that's what I got. Also, I have this little note right here. I also noticed the times on the x-axis, and again, they also give us the species richness, and if we connect it to the previous or the very first picture that we look at, it's the same thing. It looks like this right here have a higher species richness than the subfamily of the second one.
Mm. Oh, why do you think that would be? Like, why is there more richness if the first family than the other ones?
So from what I get is your question would be like, why is species, I'm gonna refer to the top one is species A and the bottom one is
Okay.
species B. Yeah. So why is A have higher fitness than B?
Or wouldn't it be diversity, not fitness?
Well, it's because
richness. I mean richness.
Yeah.
Well, it's just because the ventrals, because the ones that have ventral and lateral photophores, they are much more diverse from each other because they evolve those patterns. They evolve those patterns differently from the ventral, just the ones that have ventral alone. That's why they're much higher up on the species richness number, because they're much more diverse within each other because of the lateral photophores alone, compared to the more ancient just ventral ones.
Mm-hmm.
Because basically when they evolve with the bioluminescence trait from the clade, those ventral ones, just the ventral ones alone, were basically that was the only pattern that existed up until the ventral ones evolved it differently to have ventral and lateral, effectively like making them more diverse within each other just because of the addition of the lateral.
I think we can also connect that to the functions of the photophore as well.
True.
Like like species
Yeah.
A, it uses it, or it take advantage of that lateral, and then it can use that to communicate, you know, and that's
Yeah.
how they comes to at a much faster rate at diversify compared to species B. They may or they may not use that advantage of having that psi photophore.
Yeah.
I mean,
Okay,
and then
so
we kind of already like unintentionally discussed
yeah,
the conclusion a little bit there. It was basically that they were that both the ones that had ventral and lateral ones are
yeah,
more species specific than just the ventral ones.
definitely, yeah.
Um, let's
Okay, so
yeah,
should we move on?
this all relates. So basically, I mean the main point that sticks out as to how it relates to our course is the trees.
Yeah.
The trees is probably one of the biggest things that stick out, just because of how we work with them.
I mean, if we can look back towards our tree from the last page, we can just take about how just the traits derive from each other. Like you see, like the bioluminescence trait derives from like A, and how it's just spreads across other species.
Yeah, I mean that's what basic evolutionary biology.
Yeah.
Yeah, okay. Before we end this guy, let's point out like a clear main finding that they did in this article. What do you guys think? Um,
So you should go into the discussion. Yeah.
Oh.
I think so my little note right here again. I I the main findings of this paper is basically how different positions of photophore can facilitate in diversifications of species richness or just species in general. That's what I get. But
Yeah,
let me know if you have anything
yeah,
else.
Yeah,
that's what.
but another finding I also did is like they found that the lateral
Mhm.
biolume played a role in genetic isolation, which means that they are
Yeah,
also more diversified.
and then
Yeah,
in this paper they also explained how or why how or where the lateral positions come from. Um,
yeah, with the looking at the clades and stuff.
yeah, like first we
Mhm.
know it start all in the ventral positions, but why
Mhm.
all of a sudden it comes to the side or the lateral position, and they did a good job at explaining this as well.
Yeah.
Yeah, I think just if we knew a lot more about certain terms
Mhm.
without having to look up every single term that we didn't know, it would all obviously be a lot more easier to understand. But even as it is,
And
Mhm.
yeah, and he also said from the results that the common ancestor of lanternfishes most likely evolved their complex body of photophore systems from the late Cretaceous.
Yep,
Cretaceous,
which
yeah.
is around 100 million years ago.
Yeah.
Definitely, that's the one. And what else can we extract from this discussion?
Was it that the 4-to-4 placement on the lateral sides of the body can identify a species?
Yeah, so I think I have the note right here too. Let's see what it right here. So this tells us that the ventral it's for camouflage, and then the
Yeah.
lateral is for communications, which is exactly what you said earlier. How some species they can detect or recognize that is their species by seeing or looking at that lateral positions of that photophore.
Mm.
And then that also contributes to the genetic isolation.
Yeah. Esmeralda, if you don't mind, explaining me about the genetic isolation real quick, that'll be great. I
Because I
I'm a
feel,
bit confused on that one. So.
oh well, I feel like that it contributes to genetic isolation because since the fish can identify themselves by the lateral sites having the photophores, that means like their species will most likely tend to stay to themselves together because they know they're like similar, which is
Oh, okay.
But I also wrote down that genetic isolation is like it's like little genetic mixing with other organisms with the same species.
So if correct me if I'm wrong, but if we tie this back to the stuff that we just learned like a couple of days ago regarding like isolations, um, can we also say is that these can also speed up their speciations as well? Like over times, only that species will recognize each other, and then those species that don't recognize each other, they'll become another whole species.
Well, in my notes somewhere, I wrote that genetic isolation doesn't often result in speciation. It like rarely
Mhm.
happens.
Oh, okay.
Yeah.
It's rarely.
Yeah, it rarely happens.
But in this
Mhm.
case, it does.
Yeah.
Okay. So what would be the most common what would be the most common effects of genetic isolation?
Which is I feel like it's more like immediate communication, I would say. Since I mean, if you're more diverse, it'd be a lot more easier to just recognize, like just communicate better within your
Okay.
species, if that
Yeah.
makes sense. Just like I mean, I just in a sense like we do, we're so
Mhm.
like accustomed to each other, we're so like unique, to put it in a sense that we can very easily identify and communicate with each other, like immediately,
Okay.
essentially. But I think that pretty much concludes everything that we have to say.
Yeah. So I'll stop recording now.
Oh, yeah, 1 second before we end this. So, just like any other experiments or I don't know, research, um, even those they found what they found, so like a potential answer,
Mhm.
um, there's there's always gonna be many other things that could explain to why this is the case. And the researchers, they did a great job at explaining that even though they found that the positions of the photophores affects the
I mean, in the
diversification rate.
introduction, they explain a lot of stuff too, like how
Yeah.
bioluminescent pathway works through biochemistry and
Mhm.
biochemical processes. Yeah.
Yeah, but they also make sure that we realize that there's many other factors that contribute to this diversity as well.
Yeah.
So it's just that bio or the photophore is one of the factors.
So there
That's pretty
could
much
be
that.
other ones that can play a role in this, but like they don't really deeply go into it.
Yeah, yeah. In this paper, they just focus on that photophore position.
Yeah.
But, I'm pretty sure in the later on they may like later research or the next topic. I don't know. They may identify other factors that contribute to this as well. Yeah. What would you guys do for your follow-up experiment?
I don't. I mean, if I was in that field, I would look at
Mm-hmm.
the lanternfish with like I don't know what specifically about them, but it's really cool. The ones that like the ones from Nemo. You know, the ones that have like the
Oh,
hanging,
yeah.
The
the hanging.
anglerfish.
Yeah. I mean,
Yeah.
I know it'd be really hard to collect anything on them just because of how deep they are, but
Yeah.
Yeah.
maybe look at diversification within them and how they evolve
Okay.
that specific trait. You know, something along those lines.
Yeah.
For me, I would like I don't know, narrow the research down and maybe potentially trying to find out what are some other functions of these photophore. Like, we saw that they found two functions of it, but then I
Yeah.
don't know, they may use it for another functions. I don't know. So I would do the next research based on that.
Okay, we're approaching 30 minutes, so we should
Okay.
probably.
All right. I'll stop the recording right here then.
Okay. All right.